Obesity can cause many different diseases such as high blood pressure, diabetes, and cancer. The trillions of microbial cells in the intestinal microbiota can contribute to obesity by increasing energy extraction or by altering metabolic signaling and inflammation. Overweight and fit humans differ in their microbiota compositions. Because early life is a critical period for metabolic development, disruption during this window could lead to changes in body composition. In humans, early life microbiota disruption, either due to delivery by a C section or antibiotics, is associated with increased risk of overweight status later in childhood.
So, what is the article saying? Well, the article suggests that exposure to antibiotics during the last week of pregnancy or during nursing changes the bacterial landscape of the gut and alters the body's metabolism. This causes a predisposition to obesity. The study administered water with low doses of penicillin to 3 different groups of mice. The first group received the antibiotics during the last week of pregnancy and continued the medication throughout life. The second group received penicillin after the mice is done breastfeeding and continued it throughout life, and the third group received no penicillin. Overall, the study showed an increase in fat mass in both groups that were administered penicillin. However, the fat mass was significantly more in the group that was administered penicillin during the last week of pregnancy. With this, it was shown that the mice were metabolically vulnerable and ended up being more susceptible to obesity and metabolic problems if they were administered antibiotics early in life.
With their previous work, the authors showed that early life subtherapeutic antibiotic treatment increased fat mass and altered metabolic hormones, hepatic or liver metabolism, and microbiota composition. They decided to extend their studies using low-dose penicillin, or LDP, as a model agent disrupting the microbiota and examined the effects. The researchers first wanted to see whether the timing of LDP exposure was critical, if early exposure would have more substantial effects. They found that when you perturb gut microbes early in life among mice and then stop the antibiotics, the microbes normalize, but the effects on the host metabolism are still permanent. This supported the idea of a developmental window in which microbes participate. As shown, the penicillin did not diminish bacterial abundance in the gut. The 4 distinct organisms included were found to be 4 potential new candidates as promising probiotic organisms, so theoretically they could be given back to organisms after antibiotic treatments. Secondly, the researchers wanted to know whether synergies existed between penicillin and dietary effects. In other words, were there additive effects of early life LDP exposure in increasing the mice's caloric intake? They found that when they put the mice on a high calorie diet, they got fat, and when they put the mice on antibiotics, they got fat too. But when they put the mice on both antibiotics and a high fat diet, they got very, very fat. Normally, adult male mice carried 3 grams of fat. The animals in the study fed the high fat diet had 5 grams of fat. By comparison, the mice who received antibiotics plus the high fat chow packed on 10 grams of fat, accounting for a third of their body weight. The treated rodents were not only fatter, but they also suffered elevated levels of fasting insulin and alterations in genes related to liver regeneration and detoxification, and these are consistent with metabolic disorders in obese patients. Lastly, the researchers wanted to know whether the metabolic changes were the result of altered bacteria or antibiotic exposure. They addressed this question by transferring bacterial populations from penicillin-exposed mice to specially bred germ-free and antibiotic-free mice, starting at 3 weeks of age, which corresponded to infancy just after weaning. The researchers discovered that mice inoculated with bacteria from the antibiotic-treated donors were indeed fatter than the germ-free mice inoculated with bacteria from untreated donors. This showed that the altered microbes are driving the obesity effects and not the antibiotics.
So, how is this article related to what we learned in class? Well, there are several connections we can make to the modules we've covered. For one, we learned about the importance of the gut microbiome. Our gut microbiome helps with the digestion of certain foods, production of certain biomolecules, and overall maintain a state of homeostasis in our digestive system. The large intestine contains anywhere between 300 and 500 species of bacteria. We also learned about how hormone imbalances can cause obesity. Remember the hormone leptin? We learned that in mice that have a defective gene for leptin, which causes appetite suppression, become obese. From this picture, the mouse on the right is the control, and the mouse on the left has a defective leptin gene. Now, with the knowledge from this article, we know another potential cause for obesity is early antibiotic usage. From this article, we realize that the critical period of development extends not just for our mental capacity, but also for our metabolism. While the study doesn't necessarily have direct implications for humans, it just goes to show how important the millions of bacteria that grow inside our body are for our daily health.